Welcome to Pharmacognosy Metabolic Engineering, the NPTEL online certification course. So, now we will go to the lecture 17, where we will talk about benzylisoquinoline alkaloids, induced top 1 mutant and natural T mutant, and then once we finish this, we move into the engineering these pathways for manipulating the alkaloid levels. Although in the last class, that is class number 16, I have discussed about this top 1 mutant, but what I feel that there are some important issues which I did not discuss to that extent. So, I will cover it now. So, let us go to the next slide. So, revisiting top 1 mutant and natural T mutant of opium poppy, and then metabolic engineering of isoquinoline alkaloid biosynthesis. So, these are the two things which I intend to cover in this class. So, what yesterday I have discussed about the discovery of top 1 mutant. So, the top 1 top 1 mutant of opium poppy. So, this mutant basically was created by induced mutagenesis that I have mentioned. The seeds were treated with the mutagen and subsequently they have screened. So, one interesting aspect what they found it very useful is to find the proper mutant is that the latex of top 1 mutant was found to be colored. Top 1 mutant was colored. So, latex of top 1 mutant was found to be colored and that is yellow in color, and subsequently what has been found that this latex was dominated by the alkaloid which is thebaine. Now, next question is this, that I have mentioned about two varieties. One is the Munich variety, another one is the Tasmanian variety. So, the Tasmanian variety was used. So, the opium poppy seeds from Tasmanian variety was mutated and finally, they have recovered the top 1 mutant. And what has been found in top 1 mutant when they analyzed the alkaloid, they found that this mutant accumulates thebaine and oripapine, not the other downstream metabolites. That indicates that the step which converts oripapine to further downstream process, that is blocked, and this appears to be a demethylase 6 demethylase activity. So, that demethylase activity perhaps got impaired, although at that stage the activity was not properly demonstrated. Similarly, if this oripapine to morphinone, if this is a 6 demethylase, the similar demethylase also operates in the Munich variety. So, therefore, such varieties, if that is used for making the mutant, so there also this step will be blocked. That means, thebaine cannot be further converted into chorianone via nephionine. So, now what is the implication? The implication is this: in the Tasmanian island, so there are plenty of industries which are producing licit alkaloids, LICIT. That means, these alkaloids are safe and cannot be transformed into drug. So, there is a high demand for licit alkaloids, because these alkaloids can be used as a precursors for synthesis of other pharmaceutical important alkaloids. So, therefore, after the discovery of top mutant, the top mutant plants grown at large scale, and that is utilized by the industries located in the Tasmania for harvesting licit alkaloids, which is indeed very exciting. And not only that, what I have mentioned, that since it is accumulating thebaine, so the thebaine can be converted into several important metabolites. For example, buprenorphine which is basically better analgesic, which is considered as a better analgesic than morphine and codeine. Not only that, thebaine can be chemically transformed into several pharmaceutically important molecules. For example, oxycodone, then naloxone, and so on. So, there are heavy demands for this. So, therefore, if top 1 mutant is used for accumulation of thebaine, so this thebaine can be utilized for producing these compounds, and there lies the implication of this work. So, now let us go to the next aspect. So, this is what I have mentioned yesterday about the elucidation of the late steps of morphine biosynthesis, and there I have mentioned about dioxygenases. So, these dioxygenases plays important role. Although this is demethylase, but the function is like dioxygenases. So, that is why it is called dioxygenases. Now, what I have not specifically mentioned, that how they have, what I said that they have used the functional genomics approach. Now, interesting point is this, if you if we compare with the top mutant. So, top mutant is an induced mutant, it was not found in the nature. So, it was induced. Here they have discovered a mutant which is called T mutant. So, T mutant found in nature, and the other one which is normal wild type, that was called as L type L. So, T mutant, what they found, that which is discovered from the nature, that there it is not producing the morphine and codeine. That means, here also the steps for synthesis of morphine and codeine was found to be impaired or not functional. So, subsequent what they have done. So, next what they have done is basically they have done the transcriptome analysis from T mutant and from wild type. So, comparative transcriptomics data was used to fish out the real where lies the defect, and when they compared the transcripts, they found that in the T mutants there are genes which are not the same as they found in the wild type. That means, there lies some defect or some mutation happened, and subsequently through further analysis they have found out these two. One is that T6 ODM which operates in both the varieties. For example, in case of Munich varieties it follows this route, and in case of Tasmanian varieties it follows this route. And also so they have basically found several dioxygenases, and they have named accordingly DOX1 and DOX3. So, one of them is basically coding for this T6 ODM, that was DOX1, and the other one it code for CODM, which they call as DIOX3. and what I have also mentioned, that in order to confirm their function they have used virus induced gene silencing to block the steps, and they confirm that indeed these are the enzymes which catalyze the function, and when the gene was blocked by virus induced gene function. So, it did not push the pathway towards morphine, instead it is accumulating either thymine or oripavin. It depends on the type of the genes they have used. So, this is all about. So, this yesterday I did not mention about the T mutant and L. So, here I made it clear. So, where lies the difference between top mutant and T mutant is this. T mutant is basically found in the nature, and that was used for fishing out the genes for T6 sodium and sodium, whereas top 1 is basically the induced mutant, and where they predicted that this step was defective, but they did not characterize that. But the practical application of top mutant was that actually it revolutionized the industrial production of licit alkaloids, whereas morphine and codeine, these are called illicit alkaloids, because this can be converted into notorious drug heroin, particularly morphine by acetylation, which I have mentioned in the last class. So, one of the previous classes when I discussed about the isoquinoline alkaloid pathway, I briefly mentioned about berberine and sanguinarine. So, these are the two alkaloids. So, sanguinarine is basically a benzophenanthrinidine alkaloid, which is produced from scoularin via protropin. So, protropin group and benzophenanthrinidine group, and the other route from scoularin, it goes towards tetrahydrocolumbamine, and ultimately it forms the berberine. So, scoularin originates from reticulin, and the enzyme is BBE or berberine bridge enzyme, which I have mentioned. So, here if you see the berberine pathway, that is from scoularin, it makes tetrahydrocolumbamine by scoularin 9O methyl transferase. So, this is a very important enzyme, and then through several intermediates ultimately it forms berberine, and there is a demand for more berberine and more sanguinarine, because these are antimicrobial compounds. So, pharmaceutical industries indeed they need in more production of this, but through chemical synthesis it is difficult. Therefore, we are entirely depends on the plant. So, that is the reason scientists, once they identified the enzymes and genes, they try to manipulate the pathway, so that this production can be enhanced. So, that was what was the aim. So, keeping that in mind, we will just move into the next slide. So, this is basically the one of the first metabolic engineering applications with benzylisoquinoline pathway, where scientists made a target to express the scoularin 9O methyl transferase, which is SMT, in cell cultures of Coptis japonica and Eschscholzia californica. Coptis japonica is known for producing copticin, and Eschscholzia californica, which is called California poppy, which is known for the production of sanguinarine. Both of these compounds are of antimicrobial nature, therefore, there is a demand to produce this compound at enhanced level. So, upon expression of this SMT gene in Coptis, what is going to happen we will see, and expression of SMT gene in Eschscholzia cell culture, we will also see what has happened. So, here there are two things, because SMT is here in the 11. So, when this gene is expressed, it is possible that this pathway will be induced, and as a result of that it may lead to formation of berberine. Although sanguinarine is not meant for the production of berberine, but in Coptis berberine there are reports of berberine production, and so this will be interesting to see. So, let us see what happened. So, when the gene was expressed in Coptis cell cultures, so SMT activity was measured. So, SMT activity in the transgenic Coptis cells, what is this one, that it enhanced when you compare with the control. So, that is obvious, if the gene is expressed, it should make the functional protein, and then if the protein is there, the activity can be measured. And next what they looked into the content of the alkaloids. So, what they found, the alkaloid contents, for example, berberine, which is the close bar, the berberine content enhanced. So, WT stands for wild type and SMT stands for basically the gene which is expressed in the copticin. So, the berberine content, when you compare with this and this, here it is enhanced, whereas the copticin and columbamine both are slightly suppressed when you compare with this one. Same case, that of course, in the when the cultures were grown for 15 days, the transgenic cultures accumulated more berberine, and the content of the copticin got further suppressed as compared to the wild type. Wild type more or less it is same. Okay. So, that is quite clear, because when SMT is induced, the pathway as a new branching started, and as I said that there are reports of berberine from the coptis. So, therefore, putting an SMT gene will move the pathway in this direction, and that ultimately leads to the accumulation of berberine in higher amount in the coptis. Now, let us see what happened in the Californica poppy, that is Eschscholzia californica. So, there when the coptis SMT is expressed, so here the dotted lines basically the wild type, this is wild type, and the solid line is basically the transformed. So, what has been found, if we particularly see the first one, we see there are lot of changes. For example, in wild type there are two peaks which are where intensity is more. Now, if you see in the transformed or transgenic line, this is got suppressed. Okay. But also that there are few new products which are detected in the transformed lines, but not in the control. So, at that point of time it was not clear to them what are these compounds, but definitely upon expression of SMT in Californica poppy, some metabolic perturbation has happened, as a result of that the cell cultures have extended its plasticity, leading to the formation of several new compounds. Now, if you see these two one, so the one in the middle one B, which is basically the columbamine. So, columbamine, as I said that columbamine content has enhanced, because what I said the solid line is particularly the transformed, and this one minor one, this is what is the this is what is the control. So, compared to the control, the columbamine content has enhanced, and so let us see where columbamine lies. So, here is that tetrahydrocolumbamine. So, when SMT is expressed and scaluladin is a common substrate, so it leads to more accumulation of this tetrahydroxycolumbamine. Okay. And then sanguinarine in the wild type, that is the non transformed, this is the peak intensity of the peak, whereas in the transformed that content is suppressed, and that is expected because the sanguinarine move in this direction. So, when the pathway moves in this direction, definitely there will be shortage of substrate, and if the gene is expressed under the control of a strong promoter, so all substrates will be pulled towards this side, leaving limitation of the scaluladin for making sanguinarine, and that is exactly what happened here. So, this was basically discovered by the Sato et al. From Japan, and they published this paper in the Proceedings of National Academy of Science in USA. And much later this group, what they have done, they revisited the experiment once again, and with the availability of sensitive detection limits, what they have done, they at this point of time they characterize those unknown peaks, what they found in 2001. In 2010, when they repeated the experiment, that means they express the Coptis SMT in Eschscholzia, and that leads to formation of new compounds, which I am coming to the next slide. Just this one is just briefly showing that this is the SMT, the target one. So, this leads to the formation of tetrahydrocolumbamine, what we have seen in the other one, and this is the other pathway which leads to the formation of sanguinarine. So, what has been found here, that wild type and the SMT cells. So, in the wild type, you see these are the wild type compounds. In the transgenic it is got suppressed, whereas in the transgenic this got enhanced. In the there are new compounds which was detected in the transgenic. So, this one, this one, this one, this one, this one, and this is one is from the cell extract, that is they have extracted the cell. Another interesting point, because these are the cell cultures, there are these compounds also secreted into the medium. So, when they analyze the medium, they found that in the medium mostly the in the wild type the medium, the compounds are basically detectable. So, all the dotted lines, but whereas in the transgenic one, the secretion into the medium is less. Now, based on this, what has been found, that in the transgenic there are compounds like THC, which is allocryptopin, then 10-methyl chelidithrin, and all these compounds are detected in the transgenic cells. That means, this one, this one, this one, this one, all these they found in the transgenic cells where Coptis SMT is overexpressed. Okay. And this route is basically the controller wild type plants. So, what has been found, that when these compounds are producing in higher amounts, it also affected the wild type compounds. That means, the pathway when it moves in this direction, then definitely the original pathway is getting affected, because original pathway needs a cytochrome P450 for pushing in this direction, whereas in the transform pathway it moves in this direction. And what it indicates, that SMT and this CYP1935, these are the enzymes which involve in the reaction, and also particularly the dotted lines like this. This means that there are multiple enzymatic reactions involved in this which are not known, but one important, but to conclude this, what I must tell, that upon expression of SMT, the cell has extended its plasticity, and as a result of that, several new compounds which are not found in the wild type now produced in the transformed Californica poppy, and these compounds can be explored for new pharmaceutical applications. So, this was published in Plant and Cell Physiology in 2010 by the same group, Sato's group, and the last case studies which I will cover and end this class. This is about overexpression of Coptis 6-OMT. So, this is 6-OMT, and what happens, that when this is expressed in Eschscholzia californica cell cultures. So, other one what we have seen, basically the expression of this much late step genes, that is Scaularin SMT. So, where is which is further downstream here, but Now, they are targeting this one. So, what they have done basically they have chosen two genes, one is 6-OMT, another is 4-OMT. So, in two parallel system they have transferred the 6-OMT gene and another they found the 4-OMT gene, and they express these things, and then later they try to see what happens as a result of this expression on the alkaloid levels. So, what they found when they express the 6-OMT, that leads to around 7-fold more enhancement of the alkaloids as it is seen here, that is 6-OMT transformants. So, this is the whereas, when they have used only 4, so that is very less and almost it is equivalent to the control. So, that means that 6-OMT expression is basically performed the job well, and what is the reason for that? Actually 6-OMT in the plant is not that active. So, therefore, putting extra copy of 6-OMT basically pushing the pathway in this direction, and then 4-OMT naturally there, that will pushing further and leading to the product. Whereas, when 4-OMT was added, so 4-OMT, when 4-OMT only was used, 4-OMT has to do the job of 6-OMT as well as 4-OMT. So, because 6-OMT is not there, so this 4-OMT there, that sometimes convert this norcocloudin to cocloudin, and as a result of that the when it performs both the function, the efficiency is less, and as a result of that the alkaloid level is less. Now, interesting point is that if you see the control as well as transform, there is a coloration developed in the transform lines, and that could be because of this accumulation of the alkaloids. So, like sanguinarine and other things. So, so this is basically the HPLC analysis of the transgenic poppy. So, A is the wild type, which is the wild type. So, here these are all the compounds, and B is basically the 4-OMT transformants. Okay. So, here the increase is marginal when you compare these 4 with these 4 or these 2 with these 2. So, increase is there, but slight, but now when you compare with this is basically the 6-OMT, here there is a huge enhancement of 2 as well as 3 and when as compared to even 4-OMT and the control. So, that means the benzophenanthrenyl alkaloid content in transgenic poppy has been enhanced as a result of expression of 6-OMT, and this is a very positive result, and this was reported in the famous journal Plant and Cell Physiology by another Japanese group in the year of 2017. So, with this basically what I will conclude that the benzophenanthrenyl alkaloids can be enhanced in the cell culture of Escherichia californica by putting the Coptis 6-OMT gene. Putting a Coptis gene is a heterologous source. So, there are less chances of gene silencing, and what we saw that it worked very well, and ultimately it produces more amount of these alkaloids. Whereas, 4-OMT actually when putting 4-OMT it did not function so well, because the 6-OMT as such is feeble. So, 4-OMT had to do the job of both, and that is why the performance appears to be lower, much lower compared to the 6-OMT. So, this is all about for today this class. So, in the next class we will see other metabolic engineering applications of isoquinoline pathway. Thank you.